all of you for the second day international webinar Doctrina 2. So today's same kind of presentation related to coronavirus. So our resource person is Anurag Srivastava. Our resource person is Anurag Srivastava. So he is currently doing his PhD in University of Turin, Italy, and his topic is immunotherapy of pancreatic cancer. So he has received multiple scholarships. To mention a few, Erasmus scholarships in 2013 to 2015, NIBN scholarship 2016-2017, Borso del Recta scholarship in 2018, UNITO PhD fellowship in 2019. He has a lot of invited lectures in different universities of world and India, and he has worked in different countries in Netherlands, Germany, Israel, and Italy. So I welcome the resource person, Anurag Srivastava, for delivering today's session. Anurag, hello.
Yeah, thank you, sir. Yeah, thank you, sir. Can everyone hear me? Okay. Yeah. So thank you, Samsudin sir, for giving the introduction, and I'm glad to present the recent scientific update on coronavirus, and especially after yesterday wonderful talk by Dr. Libin. And I would like to be at that, like, match that level. And today, since I'm gonna talk about a lot of science, so some of you might not be that much familiar, so I would try to make it as simple as possible. And yeah, whenever you have any issues, you can just let me know. So I think I can start sharing the screen, sir.
Yeah, okay, it's okay.
Talking about the understanding the novel coronavirus, and I will be giving the recent scientific update. And as sir mentioned that I am working in the field of immunotherapy of pancreatic cancer. So I will be giving a detail about immunology. As the research have progressed, we have understood that this disease is more of it's not like a pneumonia, but it's more of an immune disease. So I will be talking about all those things. But before going into the details, there is some kind of issues. Okay. How to minimize this? There is a lot of trouble I'm seeing here.
Anyone? Anything?
Yeah, I'm seeing too many notifications. So okay. Can you guys see the screen, and everything is visible? Okay. So I would go with the, first of all, I would go with the basic classification of viruses. So the viruses, as you all know, that they are not actually a living organism, but it's in between the living and the non-living, and everything is based on their genetic material, whether they are a DNA virus or a RNA virus. So based on that, Baltimore classification talks about the various kind of viruses based on their genomic structure. So the first one is called double-stranded DNA virus, which is a common adenovirus, and it is also called common cold virus. Then there is a single strand DNA virus, then double-strand DNA virus. Then comes the positive single-strand RNA virus, in which our coronavirus features. And in the positive single-strand RNA virus, even the influenza virus also features in the same field. Then there is a negative strand RNA virus and single-strand RNA reverse transcriptase, which is retrovirus, in which the major virus is the HIV. And the last but not the least is the single-strand DNA RT virus. Yesterday, Dr. Libin has given very detailed introduction on coronavirus. I will just give a little bit description about the coronavirus. So I will move to the coronavirus next. So when we talk about the coronavirus, so coronavirus can be classified into different uh, four different types. So the alpha coronavirus, beta coronavirus, gamma coronavirus, and delta coronavirus. The alpha and the beta coronavirus, they infect the mammals, and they are the one who infects the mammals. And the gamma and the delta, they rarely infect mammals, and there has not been a single case reported in the human. But in the case of alpha and beta, they infect the mammals as well as the human, and in human the respiratory infection is reported in human beings, gastrointestinal in animal. So the coronavirus is, like as yesterday Dr. Libin said about that, coronavirus is mostly the main reservoir is bat. So there have been plenty of spillover from the bat to the human, but they always happen with with an intermediate host. For example, in 2003, that when SARS-CoV-1 was reported in China, the intermediate host was the palm civets, and in 2012 when the MERS was reported, the intermediate host was camel. So in the current scene of the origin of SARS-CoV virus, yesterday Dr. Libin gave a very detailed talk on this. So I'm not going to go into the detail, but the thing is, it's the natural host is the bat. The intermediate, we are not yet sure whether it is a civet or it's a mammal, it's a pangolin, or it's some other kind of animal. And then it has jumped to the human host, and now since the coronavirus outbreak, there has been a lot of hue and cry. So we have to understand very simple thing is the comparative study of all the coronaviruses which have been very lethal to humankind. So the first one was the SARS-CoV virus, which affected around 8,000 people globally, and it had a mortality rate of around 10%, and human to human transmission was very mild. The MERS-CoV virus, it affected around 2,500 people, but the mortality was pretty high, around 26%. But the point is that it had a very low to negligible human transmission. That's why MERS never became a pandemic. Now comes the SARS coronavirus 2. It has already infected around 4.9 million people globally, and more than 300,000 people have died. And the reason for this is it is highly infectious. And that's the problem we are facing now. So now I will go a little bit into the biology of the SARS-CoV-2 virus. So if you see in the structure, you see there are there are four major protein. The first protein, which covers the RNA-dependent RNA polymerase, is called the N protein, which is inside the so-called nucleus. And then you have the envelope protein, then we have the membrane protein, and the most talked about protein through which the name coronavirus has come out, that is the spike glycoprotein. And as yesterday Dr. Libin mentioned in detail about uh, uh, the 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 mechanism that the spike protein binds to the ACE2 enzyme on the plasma membrane of type 2 pneumocyte, and then that is get cleaved, and then the RNA is infected inside the virus goes inside the cell. So we talk about all these things, and then we talk about a lot about immunology, that what is the immune response, why there is a asymptomatic pressure, symptomatic patient, all these things. And since I'm going to talk about a little bit in detail about the immunological research which has been done in the past few months. So I thought that for the people who are not from the field of biology and who doesn't have any idea about immunology, I would give a very quick lesson on immunology. So I would be going with a basic concept of immunology, and I would try to explain in a layman term as much as possible. So when we talk about immunology, immunology is divided into two types. One is called innate immune system, other is called adaptive immune system. And the innate immune system is something which has evolved over the year. Like, you go to any organism, any organism in the tree of life, and you will find some kind of innate immunology. Even in bacteria which get infected by bacteriophage, they also have an immune system which protect them. So immune system is like a defense mechanism where you are like, okay, you protect the people. But the question arises, then what is there in adaptive immune system? Adaptive immune system is a pretty recent, like, in the terms of evolutionary, I'm saying that it is pretty recent because it is mostly found in the vertebrate. And even in the vertebrates, you have different things. And then it comes to the mammals, which has the most advanced immune system, and human has the even the advanced one. So here you see too many pictures in this innate immunity and adaptive immunity. The name of the cells are there in the innate immunity, the phagocyte, dendritic cells, and all these things. Adaptive immunity. Now the point is, if we have to understand the basic difference between innate and adaptive, innate immunity is a one system which just recognize a foreign element with some kind of a pattern. If I have to give an analogy, I would say that when all the students they join a college, like when I joined my college in VIT, I remember that we had a very easy time in giving proxies, because teachers just come and take attendance, and you can just say the yes sir, yes ma'am, and the attendance is given. Because here the teacher just focuses on listening. Okay, yeah, student is present. So that is like, you can cheat there. But when it comes to giving an examination, when you go for examination, you have to bring all your documents. There, doing a forgery is very difficult, because all the kind of checks are being conducted. Similarly, in innate immunity, there are pattern recognition, which recognizes, like, there are toll-like receptor which recognizes whether it's a bacterial DNA or bacterial flagella or some kind of stuff. But when it comes to adaptive immunity, this kind of recognition, the one which has come from the innate side, they help the adaptive immune system that, okay, remember these are the guys. And once adaptive immune system recognizes someone, they make a memory. So here you see, and if you see here in the timeline, the innate immunity is the first one to do the to lead the defense in the hours. Like, you have an infection and you get the response within hours. But the adaptive immune system take days to respond. But the difference between but the benefit of adaptive immune system is, once a antigen is being recognized by a naive B cell or naive T cell, they make a memory. Similarly, like, a student who has passed out from a college, while studying there, he had made some connects, like he had made some impression on the professor. So when they will go back to the same university to meet the professor, they will immediately recognize. And so when a students enter a college, at that time, the recognition of a professor and a student is pretty low. But when a student leave out, the memory has been built. So if you see, the first time the antigen is present, a first time any foreign substance is there, they will elicit a response which will be pretty low. But when a second when it comes second time, then the immune response would be pretty high because of the memory cells. The memory has been made. And this is the principle which is being used in the immunization and the vaccine development, which I will be talking in the later stage. Then yesterday there were a lot of questions asked on to Dr. Libin about the plasma therapy. So here we have here I have tried to explain the active and the passive immunity. So in the active immunity, what happens is you are infected with a foreign particle, foreign pathogen, and then after your body fights out that virus and then make a memory, and then they are ready with the memory cells. So next time it will come, it will fight it out. In this passive immunity, the thing is you take the sera from a recovered patient and then give it to the patient who is very critically ill. He will just fight it out and he will recover. So the question remains whether there is a specificity. The answer in both the cases is yes. Memory in the first one, yes. In the second one, no. Again, I will give another analogy which I have done it myself. So in my school, whenever I have an opportunity to sit behind a topper student, I wouldn't hesitate in copying his notebook, because I know the answer what he is writing is almost correct. So if you ask me after the examination what I wrote, I will be blank because I have just copied it. But when I study for a particular thing, even after the examination, I can tell you the answer. Oh, you know, I wrote about this thing. So in the first case, I was just passively copying the answer without even thinking, because I know that this is a topper guy. He is not gonna bluff, and his answer is gonna be perfect. So that is a passive immunity. And when I prepare for examination, when I prepare for a subject, then it is an active immunity, and I will have a long-term memory for that. Now, so this is a lot about the this is about the basic concepts of immunology. Now, how our immune system responds when there is a viral infection. As I told you earlier, that there are various innate immune receptor, and one of the most common immune receptor is called the toll-like receptor. So these toll-like receptor, they recognize, they are like around 8, 9 or 10 are there, and they recognize a specific pattern. So whether it's a DNA, whether it's a single strand RNA, double strand RNA, they have been bestowed with these capability. So once this uh, virus has infected our cell, it will be recognized by one of the toll-like receptor, and it will activate. There is something called interferons, and these interferons will go on to express a lot of type 1 interferon. Interferons are the one which are the major barrier against the virus fight. And if you see, toward the end, they make a antiviral state. So what happened in the if we go into the detail, so the type 1 interferon get activated. They go through a lot of signaling. I'm not going into the detail of the signaling pathway. And toward the end, they either inhibit the protein synthesis of the virus, or they degrade the viral RNA, or they inhibit the viral gene expression or the virion assembly. So interferon is a major player, and that is why we get so many infections, so many inflammation or the symptoms which we get, like a fever. One of the response of interferon is it increases the body temperature, because body temperature reduces, make the environment very susceptible for the virus or the bacteria to grow. So that's the one of the things. So these are the things. So remember that this is a production of type 1 interferon. So this is a very important player in this game of fighting viruses. Now comes the case of the asymptomatic coronavirus patient. So this is this I have taken from a paper of Nature NEJM journal, and this was the first kind of paper which reported the first European patient, which was found in Germany. And how does it happen? It happened that there was a guest from China. He had to do a business meeting, and he met few people. So that person is called as patient 1. Then he returned to China, and after I think 5 days, he started showing symptom. He came in Germany on 19th January, started showing symptom around 23rd January, and then he tested positive. That's the main patient who came from China. Now there is a patient 1 who attended the business meeting, and during the business meeting he met this guy, and during this whole stuff he's again developed after a few days symptoms and then tested positive. He met a lot of people, and these two patient 3, patient 4, and they also showed the symptom bit later. So so that's why we came to know about the term called asymptomatic carrier, that people don't even show the symptoms, but they are still the carrier. And this was something problematic stuff. And so there was a kind of a theory which was proposed that it could be a possibility that this virus is kind of not letting the interferon expression go in a proper way. Now the thing was, is it correct? Recently a paper has come out which compared the response of the cell in the influenza virus and in the SARS-CoV-2 virus. So the first slide I'm showing right now is from the in vitro study, where both the viruses were infected. They infected the cells, the epithelial, the lung cancer cell line were infected. And I would not go into the detail of this graph, this heat map and the dot plot. What like one of the thing which was very clear from this study was that interferon 1 and interferon 3 were undetectable. There were very small subset when interferon, as I showed you previously, that when type 1 interferon and get activated, it is induce a lot of genes which lead to a lot of pathways, and that lead to the killing of the cell, or at least showing some body response. It was a very small subset of interferon stimulated genes induced. But what was noticed well, that despite a complete lack of expression, it was able to elicit a strong chemotactic inflammatory response, which was like not in a proper way. And if you see here, this is after the influenza virus and SARS was infected in the ferret, is an animal, and then they studied the immune response. If you see in the heat map, you will see that on day 7, the various kind of inflammatory genes which need to be activated, see here on the influenza virus, it is too much red, but when it comes to the SARS-CoV-2, the expression is not that high. And when they compared this on a large, like on the large scale, so what they found was, they found, if you see on this side, uh, this is the expression of the all the inflammatory genes. The red is like way too much expressed, green is less expressed in the influenza virus. But the same thing, if you see on the other side of the SARS-CoV-2, you will see the expression is pretty low. And that kind of explain the reason why the patient doesn't show the symptoms. And if we have to go in a like a, if I have to summarize this study, so the thing is that SARS-CoV-2 infection induced low interferon 1 and 3 level with a moderate IgG response, strong chemokine expression, but it is again not a interferon expression. And this lead to low innate antiviral defense and high pro-inflammatory Q contribute to COVID-19. So this was a too much of immunology stuff. Now I would like to go to the a little bit less scientific term. Last week, I think last week or a week before, I got a lot of videos of Times Now saying that Italy has made the first coronavirus vaccine. And that's what being reported in the India Today, Indian Express, that Italian firm claimed to have developed vaccine that neutralizes coronavirus in human cells. They had written human cells, but the way it was presented, it felt like that the vaccine is already there and it will be like, okay, will be available in just a week or just a month or 2 months. Because if you see that video, the video was pretty, like when I saw it as a scientific person, I didn't like the video because the way Rahul Shivshankar was explaining, oh my God, the virus has been neutralized and we have a vaccine, something like that. So much enthusiasm when the thing is something different. And that's why I thought that a lot of people don't even understand the basics of vaccine development. Everyone is being asking, like one of the common question I've been asked is, oh, when is the vaccine getting developed? It's not like making a Maggie which get prepared in 2 minutes. It is a lot different than making a Maggie. So what are the things? So I will explain, like if you go into the history of vaccine development, uh, only the last vaccine, which was Ebola vaccine, came within less than 5 years. Other vaccine have taken more than 10 years. Now all the scientific community is devoted here, and we are expecting that maybe in 12 to 18 months we might see a vaccine. So here I will explain you about the vaccine development. So vaccine development has 3 phases: exploratory phase, preclinical phase, and clinical phase. So in the exploratory phase, what we do, we explore various kind of possible substances which could be used for making a candidate vaccine. And if I have to give the current scenario analogy, in the lockdown phase, I think everyone has started doing cooking. But the question remains, how many of these guys will go on to become a chef? The answer is very few. Similarly, the in exploratory phase, we explore a lot of probable combination and do a lot of like whether we have to use the spike protein, whether we have to do the like what Oxford, University of Oxford is doing, mix 2 different viruses and then try to make a vaccine which is very efficient, or RNA vaccine. All these kind of things are there. Around I think more than 100 have already passed this exploratory phase and gone into the preclinical trial. Preclinical trial include that they will, once they have a target ready, they would go on to do test it on human cell lines. Various kind of human cell lines will be tested with this. Once they see a positive result, then they will go to test the non-primates animal like mice, hamster, rat. Once the results are again promising there, they will go to do the primate animal model, which will be monkey and chimpanzee. Now I will again take you back to my analogy of people exploring food. Suppose some guy who had never cooked, now they are enjoying doing the cooking. So they will be like, oh, I think this is my new found hobby and new found, I think I can take it to the next level. So what they will do? They will ask their family member to test their food. So if they get some positive response, they might ask some friends to come over to home and then feed the food. If that is also good, then they are like, okay, I think I can now invite my little bit distant friend also for this food. If that is done, so the objective is to assess the safety and understand the cellular response, that how the candidate vaccine is acting, is able to do the is able to elicit the immune response in this in the animals. Then comes the, and I would like to tell you that around uh, around 70% of the candidate vaccine dies down at the preclinical level. Then comes the clinical trial. In the clinical trial, we have the phase 1, where a very small group of people, sometime 50 to 80, or maybe sometime it will be 20 or 30 people. This is just to test the safety, whether the vaccine which we are giving is a really good vaccine, whether you can just try it out on a large population. So this is just to test the safety of the vaccine, that vaccine is not giving some kind of responses which is critical, which is like which is hazardous to the healthy volunteers. Once this phase is crossed, we go to the phase 2, where we involve large group of people, like let's say several hundred of people, this to assess the safety, immunogenicity, and vaccine doses. And once this is also passed, then we go to the phase 3. Here we enroll 1,000 to 10,000 of people. And here what happens, this is a very important thing, that we do a randomized and double blind. Double blind means that the patient who have been enrolled, they don't know whether they are getting a vaccine or they are getting a placebo, a dummy substance, a dummy vaccine. And even the doctor who are performing, the health professional who are performing this, they also don't know whether they are giving the vaccine or the dummy vaccine. This is just to minimize the bias and to get a very rigorous and very good data. This is to check the vaccine efficacy, and especially like once in the phase 2 they found, let's say that some kind of immune responses have been generated in the body, which is not good in the phase 2, like let's say in 500 cases, one patient generated some kind of response which was not good. So they will test now by enrolling maximum number of people, maybe 10,000, 20,000, just to eliminate that this is not there. Because once the vaccine gets the approval, it will be administered to billions of people, and you cannot have a vaccine which doesn't fit all the criteria. Otherwise it could lead to some catastrophic effect. So I will again go back to my analogy of the people started cooking here in the lockdown phase. So once their family member, their close friends, and the distant friend have started liking, and the guy is also very interested in making the food for more people, so he would start like, okay, I would feed around 50 people and then take their response, which would be randomly selected. If that is good, then he will try to open a small restaurant and then start feeding, and then he will go on to become a better chef. That's how in a similar way, the vaccine also go to the phases of the trial. Currently, the WHO is updating the list of the vaccines. So there are around 8 vaccines which is in the clinical trial stage. Sorry, there are 110 vaccines which are in the preclinical trial evaluation. I'm sorry, and this is the vaccine which is the Takis vaccine, which was being reported in the Indian media as we have found a vaccine. So it is still in the preclinical trial stage. Then there are 8 vaccines which are in the clinical trial. So the most talked vaccine is the University of Oxford vaccine, which yesterday Dr. Libin also gave a little bit idea about this thing. Yesterday, I found an interesting article that the Moderna vaccine have shown very strong results. So I would first go, I wanted to start with the Oxford vaccine, but since this result have came out, so I thought I would delve a little bit into the Moderna vaccine also. Yeah. Okay, so first I will explain the University of Oxford vaccine, how it is and what it is. As Dr. Libin also explained yesterday, that what they have done is they have taken a chimpanzee adenovirus, they have modified in such a way that it is unable to cause any kind of disease, and that is used as a vector. And then from this COVID-19, they have taken out the spike protein, and then it has been mixed with this viral vector and made a, which they called ChAd-OX1 for a novel corona vaccine. And what they are doing is they are injecting it into the human. They are going to inject, and they are expecting that the antibody will be generated for these spike proteins. And once and when the person who has been vaccinated is challenged with the coronavirus, then they will be able to eliminate the virus completely. And now I will talk about the Moderna vaccine. So this is the one of its kind, because this is the first kind of vaccine which is based on the RNA. This is the mRNA vaccine, which means they have taken the RNA of the virus and then encode in a particle and then let it go inside the body. The same way the virus do the replication, in the same way this is also going to do the same replication, but it is a truncated form. So it will not make a protein it will not make the complete virus, and it will make a protein which will be able to elicit immune response. So they have found they infected 8 healthy volunteers, and in that 8 healthy volunteer, they saw the immune response, that antibodies have been generated. They collected those antibody, and then they treated then they used those antibodies on the cells infected with the coronavirus, and the cells were able to recover after those antibodies. They also did, in parallel, they also did another study, was they collected the blood sera from these patients, and also the patients who tested COVID positive but have recovered, and then they checked the immune response and the presence of the antibodies, and that looked very similar. And they have already, like, this is the result from the phase 1. They had all, like, if you remember the previous slide, it was written that they had already started the phase 2 trial, and they are expecting that by July they will go into the phase 3 trial, and where they will enroll very high number of people. And just 2 or 3 days before, the University of Oxford also published also put their preclinical trial result of research monkey on the preprint server bioarchive. The results are again very promising. What they are saying is what they did here. They took six rhesus monkey and I think 3 or 4 rhesus monkey. So 6 were administered with the vaccine, and remaining were administered with the dummy vaccine. And then both the monkeys, both the set of monkeys were challenged with the coronavirus. The monkeys which had the vaccine were able to clear out all the viruses from the lung, and they didn't develop the symptom of any pneumonia. Whereas the monkeys which were not given the dummy vaccine, they were not able to clear out the virus, and they showed the symptom of pneumonia. This is a very good news if we just take it on the face value. But there are a little bit concerned here, because in the article they speak about that the viruses were the coronavirus was found on the nasal in the nasopharynx area, which is the upper respiratory tract. So it is a bit of concern, but the thing what they are going with, they have already entered the phase 2 trial by enrolling more than 1,000 patients, where around 700 have been given the vaccine, and the remaining have been given the dummy vaccine. So it's a good news. We should hope for a positive result, because sometimes what happened that some of the results which comes into the monkeys, sometimes they get better or worse. So we have to wait for the results. So this is about the University of Oxford vaccine results. Now I will speak about some of the controversial stuff which made headline, what I would say, due to the lack of understanding of the basic science. So the first thing which made the headline was the hydroxychloroquine. It was based on this study which came from France, where they found that when they administered the hydroxychloroquine to around 30 to 40 patients, they found that it had showed some impact. And it made a big news, because the greatest leader of the world, Mr. Donald Trump, he tweeted that, "Oh, we have a miracle drug waiting to be administered to all the coronavirus patients." That was not taken well by the scientific community, because the sample size was very small, and it has never been used as an antiviral drug. It doesn't have any symptom. Like hydroxychloroquine is in the market for almost 100 years, and we symptoms. One of the it never interacts with the enzymes of the viral replication system. So a little bit confusing that how it will act. It could only act as an immunomodulator, as it has been used in various autoimmune disease, where you decrease the immune response. For that it is being used. So at that time I said that, okay, we need large sample set and randomized double-blind clinical trial. Last week 2 studies were published in NEJM and in JAMA, where they and this study came from US hospital. Again, we need to understand that in these study, it was not a randomized study, and it was not a blind study. So but we have pretty clear understanding now that around 1,400 patients were enrolled in the study of NEJM, and around similar number of patients were enrolled in the JAMA study also. What they found that if you see, especially like in the NEJM study also, they found that patients who were critically ill when they were administered with hydroxychloroquine and erythromycin, the status changes, but the patient who were administered who are not administered these drugs, their condition also improved. So you cannot say that hydroxychloroquine was the reason for improvement. And if you see, especially in the JAMA study, it was very clear. The patient who were given the drug, their condition, and the patient who are not given the drug, their condition, they remain almost same. So these studies, the large data set, like toward the end, both of these studies have asked that we should do randomized clinical trial, but the point is, it is pretty clear that it is not very effective. Currently, the remdesivir studies are going on, and we have to wait for their results also. I'm going to touch on another topic, which is which Indians took, which we Indians took on a great pride. Oh, we have the BCG. We all are BCG vaccinated, so we will not get the coronavirus, because there were certain modeling studies which were conducted by some mathematician and some biologists to say that, okay, the countries which doesn't have any kind of universal BCG program, they have a high mortality rate, but the countries which have a universal BCG program, they don't have a high mortality rate. But when I went through both these studies, what I found, it was pretty interesting, and it was again on the preprint server, not peer reviewed. What I found was very interesting, that in both the study they ignored Germany. And my question was why? Because if they would have included Germany, their model would have failed. And these are association-based study. We need evidence-based study. In order to come to a conclusion, we need some evidence proof. You cannot say that, okay, BCG could be one of the reason. And I'm glad that in India, Professor Govardhan Das of JNU, he has started a randomized clinical BCG vaccine by enrolling around 6,000 patients in Pune. And similar kind of clinical trial studies have been started in Australia, in Greece. So we have to wait for the result before we say that, oh, this is the thing which has given us the immunity. So these were the things. Now, since I live in it, and it was one of the major epicenter. I remember getting freaking calls from my friends and relatives 4-5 in the morning, because that time it was 10 a.m. in India. So everyone is calling, why you're not coming back? So I was reading about it, and then I found that there are 3 major reasons which led to it becoming the epidemic, epicenter for this pandemic. First case, as I shared you the result, as I showed you the previous result of this first Europe in Germany. So similarly, here the first case came around, I think last week of January in Rome. But that case was quarantined and was reported. The government stopped this all the flights from China. Then what happened next? What happened next is that we have a patient in the region of Lombardy. He goes to the clinic around 14th or 15th February, and he meets that I have a influenza-like symptom, and they're like, okay, take paracetamol and go back. He comes back, he goes like on a social meetings, gathering, all these things. And then he again comes back after 3 or 4 days when his condition worsened, and then he's tested positive. That guy is said to be patient 1. But the question was, he had no history of traveling to China or meeting anyone from China. So the epidemiologist in Italy coined that we never found our patient 0. And they said that the patient, what we are calling as a patient 1, could be a patient 200 or patient 400. So this was the bigger reason. The second reason, when this all happened, the Prime Minister of Italy asked that particular city in Lombardy that please go for a complete lockdown, close every social places and close the border. But the mayor of that city was like, no, we did everything right. And as it happened in India, that we fight that, oh, you are opposition party, you are saying like that, you are so similar thing happened here. That Italian mayor, what he did, he asked people to go for party and he joined them in most of the parties, putting pictures on the Facebook, drinking with these people. And after 5 or 6 days, he tweeted, guys, I am corona positive, please take this seriously. So that's what I call people's stupidity. And the third one, which is in the media reported as game zero. So there was a Champion League match which happened in near Milan. It was a match between a club from Bergamo and a club from Valencia in Spain. And in this match, around 45,000 people attended this match. And something like the game final result was 4-1. Bergamo won by 4-1. So, in a 40,000 closed environment and the virus is spreading, and it happened in the second week of February. And they were very, like, as I said, the cases were not there in the northern Italy. So people were like going freely, and 4-1 means they've hugged each other 4 times at least. And as you all know, and you must have seen those videos where you have seen that the army trucks are being asked to carry the dead bodies, or the video showing that in the newspaper that in Bergamo the number of death is so high that the arbitrary page, which used to be only one page, has gone to 10 or 11 pages. So the mayor of Bergamo coined this game as a biological bomb which destroyed the city. So these are the major reason. And another thing which why this was touted as a major reason, because after this game, players performing in this match, almost 40% of them tested positive. All the media person who covered this match tested positive. Valencia also became one of the worst affected region in Italy, in Spain. So all these things adds up. There are plenty other reason also. Like in the case of people's stability, I would say that when the cases started rising, what what government did, the first thing they did was to close the university in the northern Italy. University and schools were shut down, and students, what they thought, it is a holiday. They went to the they went to skiing, they went to beaches, they carried the virus with them now. And Italy has a very similar culture as in India, where 3 generations live together: the grandfather, the father, and the son. They all live in a close family. So when they went back to their houses, as Dr. Levin yesterday said, lot of people infected their parents and grandparents, which caused their mortality because of this comorbid nature of their waning immune system. So these were the reason I thought that I thought I would talk about it, because I have been asked so many times why Italy. And you also need to understand, Italy was the first country to ban all the flights from China, but unfortunately Italy is part of European Union, and the Schengen border were never closed until the lockdown was introduced. So the people who were in Germany can travel to Italy. So many theories were there. So this was about my talk, and I thank you all for your kind attention, and please ask your question, whatever you have. I think I can stop the sharing. How to stop the sharing? Hello.
Hello, Anurag.
Yeah.
Hello.
Yeah.
Thanks for your excellent presentation. Very fine and very informative. And already, as I mentioned, questions, queries can be answered from the chat box.
Okay. I have to follow
Yeah.
the. I don't know. If you have guys question, can you just uh, uh, uh,
Yeah,
I can't
please. Uh,
I can't see it. I'm unable to see this thing properly. I'm using it for the first time, so
okay. I'll see. Let's see. Just wait.
two new messages. This was thank you. Could you please elaborate a bit on convalescence and its effectiveness in treatment? Okay, so I think this is a question from Yashashwini Narayanappa. Okay, so she's asking about to elaborate on convalescent plasma therapy. So this plasma therapy is not a new therapy. It is a pretty old therapy. It is one of the oldest way to treat patient. The thing is, once a person has recovered, he has produced antibodies, and this antibodies could be taken from their blood and could be administered to the critically ill patient. Now, this is a positive sign, but what is a negative thing with this? This therapy cannot be scaled up. Like one person can donate maximum to 3 or 4 person, and you need something which can be scaled up. And another thing which you need to understand, till now you have seen all the positive results, but we don't know whether the result is because of this the plasma therapy which we are doing, or it is just happening because the placebo effect could also be there. So in order to make it as ICMR pointed, I think few weeks ago, that this is not a given trial, this is not a prescribed treatment, but for critically ill patient we are doing it. So that is the thing.
And one question, effect of temperature on coronavirus by Dr. Desai. The effect of temperature on coronavirus.
Yeah, effect of temperature, it's a widely speculated stuff, and that with the temperature the virus will go away and all these things will happen. And I would refer to a paper, an article in Science which covered it in quite detail. The thing with the other coronaviruses like SARS-CoV and MERS-CoV, what was there, that they didn't lasted more than few months. It has already lasted almost 6 months. If we take it from December to we are around the end of the May. So we don't know yet whether this virus can sustain the harsh temperature or not. People, when they talk about temperature, one of the things they bring it out is, oh, the influenza virus doesn't go into the summer. Yeah, that is there, but it doesn't mean that virus doesn't infect you in the summer. For example, the polio virus, before the vaccine was made, it was taken as the most dreaded disease caused by virus in summer. So we are not yet sure whether the temperature has any impact or not. Till now, I don't see that, because if you see in India, the temperature has already gone beyond 40, but the number of infection are not reducing. The number of infection is still 4,000, 5,000 every day. So it says that we don't have we cannot relate the temperature and the virus. It could be a possibility, but we are not yet sure.
There is a chance for a second attack of corona to the recovered person.
No. So this has been proven. Like South Korea was the country which South Korea and Japan were the country which said that they found COVID positive in the people who had recovered from corona. What happened normally is, sometime even when you recover from it, some traces of RNA remain in your body. And RT-PCR is such a sensitive technique that it can detect those things. And these people who, like, till now, the study shows that those who are being said that they had been tested positive after being recovery, they when they took their sample and tried to infect the cells, it was unable to infect the way it can the way it can infect it is if it is taken from a person who has not recovered. So the virulence goes very down, and it is not it is just the rudimentary thing, and it happens in lot of diseases where you have recovered from that disease, but that kind of symptom you can that can be tested positive even if you have recovered from that disease. So that is the current understanding of science. So we are not, till now, based on the scientific evidence, I can say it doesn't infect again. But again, I am saying it is based on the evidence which is till now. It could change with due course of time.
And food habits, role of food habits, whether
Yeah, because if you see the mortality rate in US, around 30% of the people who have died have been linked to the obesity. So the obesity is coming out to be a major reason. Now the question is why? So because what happened when a person is obese, in obesity there are a lot of pathways which goes under, and it has been shown that even patient with other diseases having obesity, because obesity kind of reduces your immune response. And I would like to talk about something which was asked yesterday about the traditional therapy for the immune booster. And yesterday, Dr. Libin tried to explain that thing. I would just like to add some point. So I used to in the past few weeks, I have got so many messages like this, that eat this and you will either get cured from the corona, because these are the like turmeric and all these things. When we talk about the healthy habits or the culture of adding all the things which supplement your immune system, it simply means that following a healthy lifestyle improves your immune system to work better. Just like if you have to study for something, you need your brain to be fresh. Similarly, your immune system need to be fresh. It should have all the energy, because remember, energy is something very like immune system works on a very high amount of energy, that is ATP. And that comes when all your cells are functioning properly. And for that, when you take a healthy diet, that improves the cell longevity, and the immune cells also work well. One other thing which is being said again and again, we have been taught in our colleges when we studied immunology, that sleeping properly is a good way to cure a lot of your disease, because your immune cells start functioning well. So these things, what are being said as immune like that, they will help you, but don't expect that you take it and tomorrow you will get recover. This is the major problem. People take this like, okay, we take turmeric today, okay, let's eat 200 grams of turmeric and we will get cured. No, it will cause more problem, because your body is not used to that kind of amount. So anything which you take in a regular habit, it will make, like, for example, people even question sometimes yoga and exercises. Like, you do yoga, and there is a pranayam of kapal bhati to cure your to protect your gastric problem. But let's say you have some kind of gastric problem, and today you want to do the kapalbhati and you want that tomorrow it get cured. It is not gonna happen. The same goes with the immune boosters. So you have to be very precise. Like, you can eat whatever you want, but focus on the food which is healthy in general, and don't start eating too much of those healthy stuff, because they will become harmful now. So that is what I wanted to add. Is
Some
there any information on secondary infection? Secondary infection, I think I have already explained this thing, that a person who has recovered, till now there is no report, but we have to wait. But like the science of diseases is pretty old, and with this we can say with kind of surety that there will not be a secondary infection kind of a thing, because our immune system is not like our political system, which you cannot trust. But you can always trust your immune system, because once they recognize a antigen and they produce a proper antibody, that will remain with you to fight it out. That's why the vaccination has worked till now. So in the case of the vaccine is getting developed, unless this virus mutate, which like yesterday also people were asking about the various kind of strain and all those things. There have been some kind of mutation which have been reported, but the mutation is very low. The rate of mutation is very low, which is very important for a vaccine development. If the rate of mutation is pretty high, I would quote a study from Johns Hopkins, which checked around 1,000 patients, and they found less than 1% of mutation in those viral strain. So the mutation rate is very low. So we should hope that the mutation doesn't go high.
So people are infected after 28 days, like that.
Yeah, that is what I said. The thing with this coronavirus is most of the people remain asymptomatic, because, as I explained, the interferon is the one which fights the virus mainly. So what happened is the virus is going in your body, and it is just simply residing, doing slowly replication, and slowly infecting you. By the time by the time your immune system wake up, and it's like, oh, there is some problem. So like, there are two theories on the asymptomatic stuff. First is this. The second theory says that like, if we are talking too much about the disease, but we don't talk about the recovery is pretty good. Now we have cases like 4.9 million. Out of these 4.9 million, around 80% of them get cured by staying at home. They have not been admitted to hospital. So we need to take this as a positive ray of hope. So what does it mean? That it means that a patient and it means that the patient doesn't have this like, they don't get a very high symptom, they don't go into very serious cases. And then we know that antibody based testing is not that efficient. But if I take a report of from New York, when this report came, the number of cases in New York were around 300,000. The population of New York is around 83 million. And in that report, they found with the antibody testing that around 20% of them tested positive. If I take an error rate of 5%, let's say 15%, then also we have more than 10 million of people tested positive who never showed symptom. And our antibody test will only work when you have already produced the antibody against it. So the second theory of asymptomatic goes like this, that you might have a symptom, like let's say you wake up today and you are sneezing 3 or 4 times, or you are feeling feverish. The first thing which we do, we don't go to the hospital directly. We say, okay, maybe something is wrong, maybe let's take rest for a day. That could be the symptom, that could be your immune system telling you that you have got an infection and we have cured it. So that's the second way of saying asymptomatic. So we have to wait for more science to work on this and see some good result. So this is the if. Wait, there is a question. If an undiagnosed patient, sir, you said vaccine development will take at least an year.
Yeah.
Uh, can you read that question? Yeah. So can we predict till when the effect of COVID will last? I didn't get the question. You said vaccine development will take at least an year. So can we predict till when the uh okay, so if you are asking that till when the COVID-19 will last, we have no proper answer. And yesterday I was following a report. People ridiculed Sweden because Sweden was the country which went for a herd immunity mathematical model to them wrong, and the number of death is around 2,500, something like that, and the cases are not going very far. So we don't know which is a good measure, which is a bad measure. So maybe we may get the herd immunity with due course of time. And frankly speaking, like, this will be a kind of a controversial statement, but frankly speaking, this disease is not that deadly affected. When I give you the example of the New York study, that around 20% have been tested positive. So let's see the data. On that day, when the result came, the number of tested positive with the RT-PCR was 300,000. Number of death was around 20,000. This sounds scary. Convert that data. 10 million positive, 20,000. Doesn't sound scary. So the thing is, when the whole thing came to Italy and we were forced to be in lockdown for almost 2 months, we used to have our lab meeting, and my professor used to always say that assume that you are a COVID positive. Everyone should assume that they are COVID positive and they should take all the precaution. So I was doing that. I avoided going to the shopping, only went once in 2 weeks, took proper precaution, because I am like, if I have the virus, I live alone. But if I go to the shop and during that time, if I meet, like, if I buy like without any symptom and something like that, I spread the virus to an old person who is standing even a meter away. I don't know whether if I've infected that person, and that could have lead to the some serious stuff. So that's why I avoided all these things. So this was one of the instruction given by our university, by our professors, that assume everyone is COVID positive and then behave like that. So that is how we have to behave. Like, we have to change our working style, lifestyle for some time. And it is not that deadly as such. That way MERS was deadly. MERS killed around 36% of people. But this is the beauty of the nature. Nature balances everything. So when a virus, which is a very deadly virus, it has a very low transmission from human to human. And when a virus is less deadly, it has a high transmission. Because I follow the evolution theory. So this is like a proper balancing kind of a thing. So this is what I feel about the COVID. If any pregnant woman get affected with COVID and say recovered, so would her child get in any innate okay, so it's not the innate immunity, it's the passive immunity. So if a pregnant woman get infected, it is pretty sure that she would be able to transfer those antibodies to the child as it is being transferred. A lot of like, there are very so the child in the during the pregnancy gets a lot of antibodies from the mother blood when they are inside the uterus. through the placenta. And once they are born, they get they get a high amount of antibodies, whatever antibodies the mother has produced through the breast milk. So that's the thing. So so talking on the scientific basis, yes, it would be there. We don't know. So like science talk on the evidence. So based on till now the evidence of immunology, yes, it should be. Any other year?
Can other mammals be the carrier of coronavirus? Can
Sorry,
other mammals be the carrier of coronavirus?
I didn't hear you, sir. Can you repeat the question?
Can other mammals be the carrier of coronavirus?
Okay, so the thing is, there have been reports of in US tiger being infected with coronavirus and some cats being infected with coronavirus, some dog being. So till now, the studies have shown that they got the infection from the human interaction. The other way has not been proven yet. They have tried it out, but it has not been proven yet. So the chances of you getting a coronavirus from your pet is very low, but you giving them is pretty high. So that's the thing. Okay, any other thing? Can asymptomatic COVID become fatal to the patient? Yeah, that is what I'm saying. That if let's say that if a person is asymptomatic for at least 10 days, and suddenly he start developing the symptom. As I said that that example of the patient one in Italy, he had very low symptom, and he started the symptom started getting worsen after a week, and then he was admitted to ICU. Finally he recovered, but he was tagged as a super spreader, and he infected his wife and his whole family. He worked in a company. That company had around 70 employees. All of them tested positive. He went to a lot of parties. Most of the people tested positive. So just it is a thing. It could turn out to be fatal if you have some comorbidity before. Any other questions? Why air conditioning enhances the fast spreading of virus? Uh, there is no okay. So I would go into the basics of the air conditioning and all these things, and why the temperature also comes into the play. So I will try to explain this in a very simple term. What happen is when you are when you sneeze, you you create some aerosol particle which gets suspended into the air. But when you sneeze outside and the sun is there, these aerosol particle will not remain longer because of the UV light which is coming from the sun, which will destroy all the aerosol particles. When you are inside your house, the humidity and the room temperature decide how long the aerosol particle will remain. So talking in a physical chemistry term, a aerosol particle remains for a longer time when the temperature is low and the humidity is high. So what air conditioner do? It reduces the temperature, increases the humidity. So this is what they are saying, that don't use the air conditioner, because it will increase the, like, you have to sanitize the room every now and then if you have the air conditioning. So air conditioning with a COVID patient is said in hospital not to be used. So that is the logical reason, that is the scientific reason. But yeah, the studies have not been done, but this I'm telling from the basic chemistry basis. Since bat is also from the mammalian family, is there any scope of using bat immune system to design vaccine for COVID-19? Bat could be used, but the thing with the like what you are asking is this is Shohini Kullavi, that bat is also from mammalian family. Is there any scope of using bat immune system?
System.
No, you don't design you don't design vaccine from some kind of immune system. The vaccine works on a very simple principle that you should be infected with a less virulent strain of a virus, which should be able to be first recognized by your innate immunity. We always talk about that, okay, we get the memory cell, but the memory cells come into the picture pretty late. The first role is to be played by the innate immunity. They need to first recognize them and then tell that, listen, we have an attack, prepare your arsenal and kill this guy. So then that will come into the play, and that's how the whole system will work. You cannot like replicate that bat immune system in our body. We are not going to do the engineering like that, and bat has their own way to protect it. And we have used these system of developing, like if you guys have heard about the genetic engineering term, that is by the restriction enzyme, it has been taken from the bacterial immune system, and we have used for our public well-being. But you cannot take the immune system and put it in our body now. That cannot go now. Okay, there is so many questions. Is there a vaccine for SARS? No, there was no need for a SARS-CoV-1 vaccine because it died its own natural death. It never came back. There were a lot of there is a lot of there were a lot of research went into it, but since it died its own death, so there was no need for the vaccine. Is there any study regarding the susceptibility of COVID? Yeah, so asthma patients are very susceptible to the COVID-19. It's proven, because the thing is, your immune system is already weak. When you have asthma, your immune system is already kind of weak, and when you get that thing, it will enhance it. So yesterday, Dr. Libin showed a very beautiful representation that how coronavirus kills the patients. And it is found, and it is a very interesting thing that we find that it is not the lung which is getting destroyed, it is other body organs also. So what it does, like, so I talked about interferon 1 not working well. The second thing which is being talked about is the cytokine storm. It's like, once immune system get activated, what happen is it tried to kill the virus at any cost. And in that case, it forget that it doesn't have to kill the body. Just like when you eat something and something gets stuck in your tongue, until something gets stuck in your teeth, until your tongue will take it out, it will be always going like. So similarly, the virus is an irritant to the immune system. It is going and killing it, and the virus is moving from, like, it is kind of understood that it moves from the body part, and that's why you have seen that kidney damage have been reported in the death, brain damage have been reported in the death, because the immune system is attacking everything, whatever it is saying, it could be having a virus. So asthma patients are advised not to go out just because they are more susceptible. How the herd immunity works. So if you have to say about how the herd immunity works, you take the example of influenza flu. Once a pandemic happens, so pandemic is a disease which has spread globally. Then it comes and infect everyone. There will be some who will not be able to survive, and there will be some who will be able to survive. Those who will survive will have immune system, like antibodies will be produced against it, and then they will be meeting other people, and slowly the virus will spread in everyone. So that's the way, and everyone will get some kind of immunity. That is called the herd immunity or the community immunity. And then the virus will become like an endemic. Like we will start living with the virus the way we are living with the influenza virus. Very few people know this fact, because when Italy was being hit severely at that time, I think around 10,000 deaths had happened. I made a video and I explained that in 2016, influenza storm, around 6,000 people died in a month in Italy. So Italy, why all these happened? Because Italy had a very high population of aged people. Around 25% of population is aged above 65. So these are the things. So herd immunity will go into the younger generation more than the older generation. So that's why herd immunity was not being taken as a very serious stuff, because here you are putting risk to the life of human being. So herd immunity was discouraged. But with with time, when the virus will go down, it will slowly become less virulent or something like that. We don't know. Yesterday, Dr. Levin spoke about this Spanish flu second wave. So we have to wait. Okay, what is the chance of creating vaccine for corona 2? Said it is said that both have one and two. Yeah, well, the chances are pretty high. It is Ambika Devi. Yeah, Ambika Devi has asked this question. So the chances are high, as I have already spoken about the results of University of Oxford vaccine and Moderna vaccine. And yeah, both the genes are almost 76% similar. So the Oxford vaccine started making it once the gene sequence were available online on 11th January, and I think they started around 13th or 15th January in their lab. So that's the thing. What is the effect of normal antiviral drug on coronavirus? How many strain of the virus exist in the world? So first of all, there is a confusion like antiviral drug. There is nothing called an antiviral drug. Antiviral or like there is a term called antibiotic, which is used against the microbes. So when you take antibiotic, antibiotic kills that particular bacteria. There, in the history of medicine, there is no single drug which can kill a virus. So you need to make this thing clear. There are drugs which interfere the replication of the viruses, which inhibit the progression of the virus, but they don't kill the virus. So for example, remdesivir is one of the drug, and in a recent clinical trial done more than 1,000 patients across the globe, they found that around 31% of patients showed significant increase compared to the patient who have not been administered the drug. So we have a strong hope. We hope that this works well, and we have to wait. And yeah, so this is the thing. Caesar, okay. What about the response of diabetic patient to coronavirus? Again, the diabetic patients again have a lower immune system, and reports have come out that diabetes patients get have high mortality rate compared to the non-diabetic patients. So you have to take care of all the diabetes patients. And for example, another interesting thing in the corona study, you have found that the mortality is more among male than female, and this is unresolved stuff, that why it is there. So yeah, so there are certain things like people with higher age. Okay, the present situation at least quite calmed down. The number of cases is around 800, 900 every day, and the lockdown is almost lifted from yesterday. I think even the restaurants and bars have been opened, and from 3rd January they are opening up the whole country, and the travel will be free. You don't have to carry any kind of documents, but yeah, still you have to maintain the social distancing. I'm still working from the home, It's almost 3 months. I'm at home. So yeah, for me nothing has changed. So yeah.
Okay, fine. So with that we can end our session. I think so.
No, there
Any?
is one last question. There is a question like there are two questions. Did coronavirus present in invertebrates? Do rats are susceptible to coronavirus?
Yes.
Okay,
Yes.
so invertebrates, there have been no reports. Like if you are talking about that mosquito can carry this virus just like the Zika virus, no, there has no report like that. And rats susceptible to coronavirus, yes, they are susceptible to coronavirus if they are challenged, and that's why they are used as a model. Because first so what we do when we do the model study is we first take an animal, inject them with the vaccine once, and after the vaccine has been injected, we challenge them with a disease causing virus, and that is how we study. What is the future of coronavirus? We don't know, but I think it will not be that deadly as it is being projected. And slowly we have a vaccine, we will have some more cure. Because we talk about the second wave, but that happened in 1900 around 1920, and that time there was no vaccine which was quite common. Now the science has developed so much, and now we are talking about that we will develop a vaccine within 12 to 18 months. So it's quite interesting time. So we have to see. So the person who are reported in not contact, it's the community transmission. Once the community transmission has started, then you cannot trace back. And the community transmission is a thing which is not the matter of if, it is the matter of when. Like the government of India is not accepting it, but we have to accept it that the community transmission has already happened, and it's not a thing which we have to fear about. It is a thing we have to accept it. Yeah, I think
Oh,
yeah.
okay, okay. With that, we can end our today's session. Nice, excellent information we got from you. And if no more questions, I would like to call Dr. Arshad, Assistant Professor, Department of Zoology, to give his vote of thanks. Arshad, sir.
Yeah, I'm here.
Okay.
Thank you, Anurag. Thank you for the nice session. So it was very interesting, and we all found worth for the time that we spent during this particular session. So first of all, I'm thankful to the organizers, the principal, IQAC, the Department of Zoology, and all the teaching and non-teaching staff of our college for organizing such an interesting and worthy webinar in this boring lockdown period. Okay, this lockdown period is worth now due to this particular webinar that we conducted. And Shamsu sir, is there anything? Okay, we'll continue. We express our sincere gratitude to Anurag Srivastava. He is also a friend of me, and we were together a long time back during our studies research at SM College, Kannur, and the VIT. So I'm thankful to Anurag Srivastava on behalf of our department and college for accepting our invitation as a resource person. And I'm thankful to our department head, Ms. Mundas, and our principal, Dr. Ashraf, for encouraging and supporting this particular webinar. And I am also thankful to Dr. Lebin, who was the resource person yesterday, and his section was good and informative, that we have covered all the we can just cover the misbelief and all those things, and we all cured from those things than the corona. Okay, and we are also thankful to the huge participation from all over the world, and we have around 300 participants, and they were actively participating through discussion and listening, and also by asking questions. So we are also thankful to those participants, and thank you once again. Thank you all. Back to moderator Shamsuddin sir.
Okay, thank you, Arshad, sir. And so with that, we are ending our 2 days international webinar Doctrina 2. So all the participants, thanks for your kind and patient listening throughout the session of the webinar. So we will send you the feedback form. So kindly please fill the feedback form. So if you are filling the feedback form and sending it quickly, we'll be later issuing the certificate within a week because of the huge registration. So thank you once more to the resource person and all of you. Thank you.